A very good day everyone. My name is Dr. Barclay Karnatki and welcome back to my YouTube channel. So in the last video we spoke about amyloidosis. In this video we'll be talking about neoplasia and discussing a few characteristics of benign and malignant tumors alike. So now to make it easier, we'll discuss neoplasia under 4 simple headings. The first one actually being what is neoplasia, the second one being the genesis of the tumors or carcinogenesis, the third one being the progression, how the tumor progresses, how it spreads, and the fourth one being a few clinical characteristics to maybe help identify between benign and malignant tumors alike, right? So now we'll start with what exactly is neoplasia. Now the word neoplasia is a derivative of the Greek language where neo means new and plasia means form. So essentially it means new form or new growth, right? And that's what cancer is. It's a new growth of cells that constitute a tumor. Now we have to differentiate this uncontrolled growth of cells versus this entity called hypercellularity. How do we differentiate between, whether it's a tumor or it's just a hypercellular mass? Now tumors, first of all, are uncontrolled, right? The body's defense mechanisms cannot control this growth, or if they do, very ineffectively. They are abnormal and they are irreversible. The main differentiating point is that when a tumor progresses, there will be a predominance of one isoform on examination of this tumor, whereas in hypercellularity, all the various cells derived from their individual lineages will proliferate equally. Now, once a tumor is formed, we have to differentiate whether it's benign or malignant. Primarily, the only thing that we look at as students in this case is if the tumor can spread or metastasize. If the tumor can metastasize to another part of the body, maybe to another organ, then it is malignant. However, if it does not, then it is benign. So now after we know what neoplasia is, just a brief overview, we will actually look at the progression of this tumor or this entity called carcinogenesis. Now, carcinogenesis, while looking at it as a whole, we can put it into two parts. The first part being the carcinogens or the causative agents causing DNA damage, and the second one being DNA mutations, which eventually disrupt key regulatory systems. Now, carcinogens are agents that damage DNA, increasing the risk for cancer. And we have a few important carcinogens, a few being alcohol. Alcohol can cause cancer of your esophagus, your gastrointestinal system, your liver. Cigarette smoke causing cancer of your lungs, of your mouth, of your pharynx. Asbestos causing lung cancer. But carcinogens are not only limited to, let's say, chemical agents. There can be viruses as well, right? Or radiation. A very classic example of a virus causing cancer is Epstein-Barr virus or infectious mononucleosis, causing cancer of the nasopharynx. Ionizing radiation, of course, is a well-known causative agent of cancer, right? Now, other than these carcinogens, what are the mutations that actually disrupt the cell's regulatory system? These include proto-oncogenes, tumor suppressor genes, and regulators of apoptosis. Apoptosis is what was discussed in the first video. So now we will look at mutations, and under mutations we will look at oncogenes. Mutations of proto-oncogenes, which is the precursor of these oncogenes, form oncogenes that lead to unregulated cell growth. They are associated with a few things, but let's not get too complicated by discussing the various growth factors and the growth factor receptors. But when looking at mutations, right, there have to be regulators of this abnormal proliferation of cells as well. And there are two very important genes that actually regulate this growth or this abnormality. Amongst many, we will discuss the two. The first one being the p53 gene. Now, what does the p53 gene do? It's very simple. If the cell divides too rapidly, that's what actually leads to cancer, right? So the p53 gene actually slows the cycle and upregulates DNA repair enzymes. So if you're slowing the cycle down, you're basically protecting the body from this abnormal cell growth. The RB gene also works in a very similar way. It actually regulates the progression of the cell cycle from the G1 to S phase. And once mutated, it releases a factor, right? And this factor is what basically protects your body from this abnormal growth rate. Now, what happens to the cells? Like, why do they proliferate so fast? Now, cells are linked together by this binding thing called E-cadherin, which is almost like a linker, which prevent the cells from going astray. The sum of all of the problems we've discussed earlier, the carcinogens, the DNA mutations, right? These result in a problem where this E-cadherin is actually not able to hold the cells in place. And if the cells are not held in place, they start spreading locally. Now, we had discussed earlier, right? The difference between a benign and malignant tumor is its ability to metastasize. So, how exactly does a tumor metastasize, right? Very common way it metastasizes is actually through blood, which is called the hematogenous route. It can also spread locally or via the abdominal cavity of the body. Now we will look at the last bit, which is basically the clinical features of a tumor, right? So clinically, a tumor cannot be differentiated 100% by just palpation or on clinical examination. A biopsy is absolutely essential in order to differentiate a tumor between it being benign or malignant. But malignant tumors usually are more rapid growing, right? Obviously, they spread to other parts of the body very quickly. They are not demarcated on touch. They are fixed to surrounded structures as they are spreading. However, as I've said, a proper evaluation can only be done on biopsy. Now, is it necessary that every single lump has to be biopsied? Not necessarily. Tumors release a few proteins called tumor markers, right? And now these tumor markers basically indicate whether this is a specific tumor or if it's just a growth. So a very well-known tumor marker is basically in prostate cancer or in tumors of a prostate. PSA is released. And this PSA, when the levels are elevated, we always suspect prostate cancer. Now, microscopic features, right? A biopsy is done, but we have to still be able to identify whether this is actually a tumor or if it's just a growth. Now, in cells where you have taken these cells from a tumor, the nucleus is always enlarged. So the nuclear to cytoplasmic ratio is actually really messed up in these cells. And there will be a high mitotic activity. Now, based on this biopsy, a tumor is staged, and this staging is called the tumor nodal metastasis staging or the TNM staging, right? So T basically describes the size and extent of the tumor. N stands for lymph nodes, which describes the affected lymph nodes, whether it's a group, many groups, the area. And M stands for metastasis, basically where the tumor has spread to, individual tissues or organs. Thank you so much for your time and attention, and I hope this video has made the understanding of neoplasia a little bit easier.